Good evening, all. Hope I am clearly audible and my screen is visible to you. So, welcome to week 10 experimental biotechnology tutorial session. So, I am Ananya Chatterjee. I am a PhD student in IISc Bangalore and I am the TA of this course, experimental biotechnology, and course instructor is Professor Vishal Trivedi from IIT Guwahati. So far we have completed till week 9, and for assignments we have completed till assignment 8, and this is the YouTube link for all tutorial classes I upload, and this is a folder link of tutorial slides. Today we'll be starting with discussion of week 9 assignment content. Yes, assignment questions which was related to microscopy. The first question is, which type of microscope is better suited for observing thick specimens like tissue culture flask or living cells in culture dishes? Upright microscope, inverted microscope, both are equally suitable. Neither can be used for such specimens. So the answer will be inverted microscope. Okay. Then, what is the primary role of the dichroic mirror in an epifluorescence microscope? So, options: to block the emitted light, to allow excitation light to pass through the specimen, to direct the emitted light towards the detector, to adjust the magnification. So, the function of dichroic mirror in an epifluorescence microscope is to allow the excitation light to pass through the sample or specimen. Next question. What is used for fixing cells for immune localization experiments? So, what are used to fix those cells? We don't use BSA, we don't use 1x PBS. For fixation, we don't use 1x Triton 100, we use methanol and acetone for fixation, okay, to reduce movements inside cells. Let me know if you have any doubt. Next question. Why is fixation of the specimen an important step in immunolocalization? Options: it enhances the specificity of primary antibodies, it preserves the cellular structure and prevents degradation of proteins, it amplifies the signal from secondary antibodies, it's not necessary in immunolocalization protocols. So why is fixation of the specimen an important step in immunolocalization? Why do we fix? So it stops all the movements. That means it preserves the cellular structure and prevents degradation of proteins. In the immunolocalization protocol, what do primary antibodies specifically bind to? Okay, the specimen, the fluorophore, the secondary antibodies, the target antigen or molecule. So answer will be the target antigen. So because the protein or whatever the target antigen is, primary antibodies go and bind to those antigens. What is the purpose of blocking in an immunolocalization protocol? To permanently attach the specimen to the slide, to prevent non-specific binding of antibodies, to increase the fluorescence intensity, to amplify the signal from primary antibodies. So what is the purpose of blocking? to prevent non-specific binding of antibodies. So this is the same reason not only for immunolocalization but also for Western blot, for ELISA, whatever the binding studies are involved, no, everywhere the reason is the same. Next question. What would be the maximum resolution that can be achieved if a sample is illuminated by green light and observed through an oil immersion objective with a numerical aperture of 1.6? So you have to calculate with a formula that the lambda divided by 1.6 into d, whatever the formula is, you have to calculate from that formula. Okay, the resolution, limit of resolution equals to lambda divided by numerical aperture. Okay, so what is numerical aperture? Numerical aperture is the refractive index into sin theta. Oh, okay, so here already numerical aperture is given, so you have to just do the calculation. So let us do the calculation. Numerical aperture is 1.6, so that will be lambda divided by 2 into numerical aperture. So the answer is 514 divided by 2 into 1.6. Let's calculate. It's around 160.6. So the option will be C. What is used? So let me write it down. Sorry. Okay. Sorry. Let me connect my pin. So the answer will be 514 divided by 2 into numerical aperture, okay, equals to 514 divided by 2 into 1.6 equals to 160. The next question, what is used to remove water from biological samples before SAME and TEM? Washing with gradient solutions of ethanol, this is the right answer. So what we gradually do, we increase the percentage of ethanol 20, 30, 50, 70, 100, and likewise we are removing water from biological samples. Which of the following is common conductive coating material used in same sample preparation, which is gold? What is the purpose of sputter coating in same sample preparation? To add color to the specimen, to make sample conductive, to preserve the specimen structure, to remove excess fixative from the specimen. So the sputter coating is basically to make the specimen conductive. In TEM, how do electrons interact with the specimen? Electrons are absorbed by the specimens, electrons bounce off the specimen surface, electrons pass through the specimen forming an image, electrons scatter randomly inside the specimen. So electrons basically pass through the sample. So this is the sample, electron basically pass through, okay. Which type of the cells allow entry of trypan blue? Dead cells. Only dead cells allows entry of trypan blue. In flow cytometry, what is the primary purpose of the forward scatter channel? In flow cytometry, what is the purpose of getting the first question is, in flow cytometry, what is the primary purpose of the forward scatter channel? measure cell density, or to analyze cell size, or to detect small particles, or to sort cells. So the primary purpose of the forward scatter channel of FSC scan channel is to analyze the cell size. And what is the purpose of gating? To select a subset of cells for analyze. So this is basically a gate to select a particular subset of cells for analyze. Compensation is performed in flow cytometry because of spectral overlap between two fluorophores. So sequencing of biomolecules, DNA, RNA. So today we'll be discussing about sequencing, DNA sequencing, RNA protein sequencing. So DNA sequencing, here we start. So this is a sequence of single stranded DNA. So here you have shown that amplification with modified bases, chemical reaction. So we'll be discussing two methods, Sanger sequencing method and Maxam Gilbert method. DNA sequencing, historically there are two methods of DNA sequencing with a similar principle of breaking the DNA, chemical or enzymatic method, into the small fragment followed by separation and analyze them on a high resolution electrophoresis gel. Dideoxy chain termination or Sanger methods. This method is originally developed by Frederick Sanger in 1977. In this method, a single stranded DNA is used as a template to synthesize complementary copy with the help of polymerase and in the presence of nucleotides. The polymerization reaction contains a primer and nucleotides, 3 normal nucleotides and 2 prime 3 prime deoxy dideoxy nucleotide triphosphates. So when DNA polymerase utilizes DDNTPs and nucleotides, it gets incorporated into the growing chain, but chain elongation stops at DDNTPs due to the absence of 3' hydroxyl group. In the typical sequencing reaction, 4 different DDNTPs are taken into the 4 separate reactions and analyzed on high resolution polyacrylamide gel electrophoresis. The ratio of NTPs and DDNTPs is adjusted so that chain termination occurs at each positions of the base in the template. Sanger protocol, step 1. A primer is added and annealed to the 3' of the DNA template. Step 2. What happens? A radioactive ATP to label the primer. Step 3. The polymerase reaction is divided into 4 reactions. Step 4. DNA synthesis continues until terminated by the incorporation of specific DDNTPs. And step 5. A chase of polymerization reaction is performed in the presence of high concentration of NTPs to extend all non-terminated sequences into high molecular weight DNA. These high molecular sequences will not enter into the sequencing gel. So this is sequencing gel, A-len, T-len, G-len, C-len. So a chase of polymerase reaction is performed in the presence of high concentration of DNTPs to extend all non-terminated sequences into high molecular weight DNA. These high molecular weight sequences will not enter into the sequencing gel. Dideoxy chain termination or Sanger methods. So leveling or chain termination protocol. This is region to be sequenced, primer annealing site. So here you have to add primer, and then leveling or termination protocol, Sanger protocol. Second, so a primer is added and annealed to the 3' of the DNA template. Step 2. A limited amount of NTPs are added along with one of the radio labeled nucleotide to level the DNA through the length. The polymerase reaction is divided into 4 reactions. Okay, the polymerase reaction continues with 4 nucleotide and 1 DDNTPs. Synthesis is terminated at the specific DDNTPs, either A, G, C, T, to give DNA fragment to different length. So you add 4 nucleotides and polymerase. Polymerase synthesis proceeds until DNTPs are exhausted. And then this is a reaction where you are using dideoxy adenosine triphosphate and other nucleotides are normal. T reaction, G reaction and C reaction, and so on. And then here Sanger protocol is that A reaction you are adding ATPs and then other nucleotides and then add chase. So this is sequencing gel. So this is a DNA sequence at A reaction. This is a T reaction, G reaction and C reaction. So wherever there will A, there the thing will get stopped. So now if you load into the gel, you will see this kind of bands pattern, and this is from the sequencing result. So this is basically the Sanger sequencing, where you basically complement with the help of complementary base pairing and with the help of dideoxynucleotides you are working this. Maxim Gilbert method. This method was discovered by Maxim and Gilbert in 1977, which is based on chemical modification and subsequent cleavage. In this method, a 3' or 5' radio level DNA is treated with a base specific chemicals which randomly cleaves the DNA at their specific target nucleotide. These fragments are analyzed on high resolution polyacrylamide gel and a autoradiogram is developed. The fragment with terminal radio level appears as band in the gel. So let us discuss Maxim-Gilbert method. The chemical reactions are performed in two steps. Base-specific reaction. Different base-specific reagents are used to modify the target nucleotide. Reaction 1. Dimethyl sulfate modifies the N7 of guanine and then opens the ring between C8 and N9, which is G reaction. Reaction 2. Formic acid acts on purine nucleotide G plus A reaction by attacking on glycosidic bond. Reaction 3. Hydrogen bakes a ring of pyridine T plus C reaction. Reaction 4, where as in the presence of salt, it breaks the ring of cytosine. Cleavage reaction. After the base specific reaction, piperidine is added which will replace the modified base. After the base specific reaction, piperidine is added which will replace the modified base and catalyzes the cleavage of phosphodiester bond next to the modified nucleotide. So interpretation of the bands in autoradiogram. So the fragment in G lane is read as G, whereas fragment present in G plus A but absent in G is read as A. Similarly, fragment in C is read as C, but fragment in T plus C, which is absent in C, is read as T. To get the DNA sequences, the band with the lowest molecular weight is read followed by the next band in the fold length. G lane. The band is of lowest molecular weight followed by the band in A lane. Overview of protein sequencing. Okay, so this is how the Maxim Gilbert works. Now overview of protein sequencing. So it's the protein. Now you are reacting the protein with FDNB and hydrolyzing it and separating amino acids. Reduce disulfide bond, you are reducing the disulfide bond if it presents. Cleave with trypsin, sequence by Edman degradation. Cleave with cyanogen bromide, sequence by Edman degradation. So stage 1, breaking disulfide bonds. The disulfide linkage interfere with the complete sequencing procedure as it doesn't allow the release of cleaved amino acid from the peptide chain. There are two approaches to disrupt the disulfide linkage in a protein sequence. In first approach, protein is oxidized with a performic acid to produce two cystic acid residues. So this is oxidation of performic acid, this is reduction of dithioddt. So now followed by after reduction, so this is dithiothiol or DDT. So after reduction you are doing carboxy methylation by iodoacetate. Okay. So in the first step, what is happening after oxidation with performic acid, two cystic acid residues are producing, and in this approach what is happening is getting reduced by DTT or BME, and then to forming 2-cysteine followed by treatment of iodoacetate to form carboxymethyl cysteine. So this is carboxymethyl cysteine to stop the reformation of disulfide bond. Stage 2, cleavage of the polypeptide chain. So cleavage of the polypeptide chain, protease and the chemical reagents targeting proteins have a specific recognition sequence and they cleave after a particular amino acids. So some common reagents for fragmenting polypeptide chain, reagent cleavage points. So this is trypsin after lysine and arginine it cuts, chymotrypsin cuts after phenylalanine trypsin tyrosine, pepsin cuts after leucine phenylalanine trypsin tyrosine, cyanogen bromide cuts after methionine. Sequencing of the polypeptide chain. So DNFB and other. So it's so what is the mechanism behind this sequencing of the polypeptide chain? So sequencing the peptides. Once the peptide fragments are generated, we can start the sequencing of each polypeptide chain. It has following steps: identifying the N-terminal residues. So the N-terminal amino acid analysis is a 3-step process. At first, derivatization of terminal amino acids. The chemical reaction is performed to leveled terminal amino acids. So you can see this is the amino acids, and then it's reacting with DF and B. So is performed to leveled terminal amino group with compounds such as Sanger reagent. So this is Sanger reagent 1-fluoro 2,4-dinitrobenzene and dancial chloride. So in most of the cases, these reagents also level free amino acids present on basic amino acids such as lysine and arginine. Dinitrofluorobenzene reacts with the free amine group to form dinitrophenyl amino acid complex. So the next step will be hydrolyze the protein. So acid hydrolysis of dinitrophenyl amino acid complex leads to breaking of peptide bond to release dinitrophenyl amino acid complex in solution. Step 3 will be separation and analysis of derivatized amino acids. So HPLC or TLC, separation of complex and comparing with the standard amino acids. Sequencing of the polypeptide chain. So IDMAM degradation sequencing method has following steps. Similar to Sanger reagent, phenyl isothiocyanate reacts with a terminal amino group to form a cyclic phenyl thiocarbamyl derivative. Under acidic condition, the terminal amino acids is cleaved from the main chain as thioazolinone derivative. Thiazolinone derivative is extracted into organic solvent and it forms phenyl thiohydrantoin amino acid PTH amino acid derivative in the presence of acid. PTH amino acid, then what happens, the amino acid complex can be identified by HPLC or TLC and comparison with the standard amino acids. So sequencing of the polypeptide chain, C-terminal residues. Not many methods are developed for C-terminal amino acid analysis. The most common method is to treat the protein with a carboxypeptidase to release a C-terminal amino acid, and then what happens, and test the solution in a time-dependent manner. Stage 4, ordering the peptide fragments. The uses of different protein cleavage reagent produces overlapping amino acid stretches, and these stretches can be used to put the whole sequence. Locating disulfide bonds. The protein cleavage by trypsin is performed with or without breaking disulfide linkage. Amino acid sequence analysis of the fragments will provide the site of disulfide bond. The presence of one disulfide will reduce two peptide fragment will appear as one large peptide fragment. Mass spectrometry method. In recent past, mass spectrometry is conjugation with proteomics information is also been a popular tool to characterize each peptide fragment to deduce its amino acid sequence. The minor detail of this approach can be explored by following articles. So this like the details, you if you want to study, you can follow this article. So you can do mass spectrometry also in conjugation with proteomics information. So this is also a popular tool to characterize each peptide fragment to deduce its amino acid sequence. So just let me know if you have any doubt. Just wait for 1 minute. I hope my screen is visible. Can you share the PDF? So now we'll be discussing blotting techniques. So Western blotting techniques we already discussed. We'll be discussing for DNA, detection of DNA, southern blotting, and for detection of RNA, northern blotting. So for northern blotting, the step 1 will be mRNA isolation. So you have to isolate your mRNA. Then step 2, separation of mRNA fragments on agarose gel. Step 3, transfer of an mRNA from gel to some membrane. Step 4, cDNA probe preparation. Step 5, hybridization with labeled probe. Step 6, washing and autoradiogram. Then material 1x SSC, which is 5 millimolar sodium citrate and 150 millimolar NaCl. 5x buffer, 0.25 molar tris ACl, pH 7.5, 0.5% sodium pyrophosphate, 1% PVP, 1% BSA, 1% Ficol, 5% SDS. Pre-hybridization buffer, formamide, 5x buffer, H2O NaCl. Salmon sperm DNA, Wattman 3 mm paper, labeled DNA, RNA or DNA probe, UV lab, shaker, water bath. Northern blotting, step 5, at first you have to isolate mRNA. So this is 5' UTR, coding sequence, 3' UTR, poly A tail. It has an N-terminal cap structure, coding sequence, and then C-terminal poly A tail. The nucleotide A forms 2 hydrogen bonding with nucleotide T, and this pairing is very specific. Exploiting this feature, mRNA population can be isolated from RNA pool using a poly T affinity column. Northern blotting, lysis or binding buffer. So this is tissue homogenization, and then that will be followed by binding. So this is a oligo DT column, so that the poly A tail will get bound to it. And then you have to do the affinity purification, that you have to do wash, then elution, and then purification. So release of total RNA either by a lysis buffer containing detergent or by homogenization in the case of hurt tissues. Mixing of poly T containing bits with a total RNA preparation. Due to mutual exclusive affinity, mRNA binds to the poly T bits. Wash the bits with washing buffer to remove non-specific cross-contamination species. Elute the mRNA from bits, and its purity can be checked on polyacrylamide gel. Northern blotting, separation of mRNA on agarose gel. So you have to run denaturing agarose gel, because RNA sometimes do secondary structure formation. Transfer of gel to nitrocellulose membrane. Place the gel in an RNase-free petri dish. Rinse 4 times with sufficient deionized water. Soak the gel for 20 minutes in 0.05 normal NaOH and keep it enough in 20x SSC for 30 minutes. Place 2 pieces of Wattman 1 mm paper for the size of gel on the glass plate and wet it with 20x SSC. Place the gel on the filter paper and remove any air bubbles trapped between the gel and the filter paper. Place the membrane on the gel, avoiding the air bubble. Flood the surface of the membrane with 20x SSC. Place 4 sheets of this Wattman paper on the same size on the top of the membrane. Place a heap of paper towels on top of the Wattman paper and add an approximately 500 gram weight and leave overnight. Preparation of a radio level probe DNA. Preparation of probe DNA. Preparation of complementary DNA or cDNA. Multiple approaches are being developed to prepare complementary DNA from isolated mRNA. In all approaches, the 3 steps are performed. First strand synthesis with a reverse transcriptase, removal of RNA template, second strand synthesis. Preparation of probe DNA. This is a poly A tail, then oligo DT. So though in the oligo DT column, your poly A tail having poly A tail mRNA will get attached, and then so what will happen then? Reverse transcriptase plus, so you are doing reverse transcriptase and then you are adding some RNase H, so that random primers and DNA polymerase. So this is basically Hoffman method. In this approach, after first strand synthesis using oligo DT primer, so this is basically preparation of probe DNA. So here you are using oligo DT not for binding but for as a primer in the presence of reverse transcriptase and DNTPs. So you are using reverse transcriptase and DNTPs because you are synthesizing the cDNA. So then DNA RNA hybrid is treated with RNase H to produce nicks at multiple sites. Okay. So then DNA polymerase is used to perform DNA synthesis using multiple fragment of RNA as primer to synthesize new DNA strand. This method produces blunted end duplex DNA product. So radio leveling of probe DNA, 5 prime to 3 prime, 3 prime to 5 prime, the lambda exonuclease you are using, then terminal transferase plus radioactive DATPs. Terminal transferase, in this method a terminal transferase enzyme will level the probe at the end to the last nucleotide of the probe. Probe is incubated with a level nucleotide, and terminal transferase enzyme will add the level nucleotide at the end. A partial purification with gel filtration column will give level primer. Step 5, hybridization. So you have to rinse the membrane in buffer and then place it on a sheet of Whatman paper, heat it for 2 minutes at full power and further cross-link with UV rays. Then you have to pre-hybridize the membrane wet in the buffer. Next you have to place it for pre-hybridization solution. For hybridization, denature a double-stranded probe by heating in a water bath for 10 minutes at 100 degrees Celsius and then by quick chilling. Then pipette the desired volume of the probe into the hybridization tube. The membrane was washed for 5 minutes in this buffer, and then air dry the membrane, and and then you have to expose this to X-ray films at -80 degree with intensifying screens and prepare autoradiograph. So autoradiogram, you have to air dry the membrane, okay, to expose to sensitive X-ray films, and then sorry and prepare the autoradiograph. Why it's becoming black? Yeah, okay. So then the intensity of the bands on the developed film, which is a measure of measure of specific mRNA in the sample, can be compared in different samples. Okay. Now we'll be discussing Southern blotting. So again, in the step 1 you have to first isolate your DNA, then you have to digest to generate different fragments, separate those DNA fragments on agarose gel, transfer the DNA from gel to the nitrocellulose membrane. Step 5, hybridization with radio level probe. Step 6, washing and autoradiograph. DNA applied to gel, then you do electrophoresis. Probe hybridized to complementary DNA sequence, exposed to X-ray. So before that you have to do the transfer, same as RNA. You have to transfer the nitrocellulose membrane, and then have to hybridize with unique nucleic acid probe, and then you will be able to see. So the process of genomic DNA isolation, you have to lyse the cells with detergent containing lysis buffer. So this is culture cell or tissue. From there you will those cells you have to lyse. You have to do enzymatic digestion, precipitation with absolute alcohol, purification with chloroform or phenol, precipitation with absolute alcohol, and solubilization in water. Incubation of cells with digestion buffer containing proteinase K SDS to release genomic DNA from DNA protein complex. Isolation of genomic DNA by absolute alcohol precipitation. Purification of genomic DNA with phenyl chloroform mixture. Chloroform phenyl mixture has two phases basically, aqueous phase and organic phase. In this step, phenyl denatures the remaining proteins and keep the protein in the organic phase. Then the genomic DNA present in the aqueous phase is again precipitated with absolute alcohol. Genomic DNA is analyzed on 0.8% agarose gel, and a good precipitation of genomic DNA give an intact band with no visible smear. Generation of suitable size fragments. Restriction digestion. Genomic DNA can be digested with a frequent DNA cutting enzymes such as EcoR1, BAM-H1 to generate the random sizes of DNA fragments. The criteria to choose the restriction enzyme or pair of enzymes in such a way so that the reasonable size DNA fragments will be generated. As fragments are randomly generated and relatively big enough, it's likely that each and every genomic sequence is presented in the pool. And mechanical shearing, genomic DNA can be fragmented using mechanical shearing also. Restriction digestion of genomic DNA. So the protocol is digest around 10 to 20 microgram of the genomic DNA with EcoR1 2 to 3 units for each microgram overnight in the presence of appropriate buffer. Take out small aliquot from the restriction digestion reaction and check on 0.8% agarose gel. Appearance of smear with visible bands indicate complete digestion of genomic DNA and the suitability of the sample for sudden blotting. So step 3, separation of DNA on agarose gel. Separation of DNA on agarose gel, you have to just make the gel and then add your sample. So in a standard gel to resolve the DNA. As a standard practice, agarose used for southern blotting is of ultra pure quality to avoid contamination affecting migration of DNA. Observe the gel in UV translumimeter and record the pattern. Then set up the blotting tray. Transfer membrane DNA agarose gel wet. This is the wet heap of blotting sheets, glass support, then glass container. So add 500 to 800 ml of transfer buffer in the transfer tray. On a glass solid support, place a piece of non-woven vapor with the size of solid support to serve as a wick. The same as northern blotting, you have to make sure that you are giving some weight to facilitate the transfer. And then place a gel on the top of the wick and remove bubbles. Overlay with nylon membrane on the gel and carefully remove the trapped air bubble. Then carefully lay the filter paper on the membrane. Remove trapped air bubble by rolling glass rod. Place a stack of paper roll over the filter paper. Place the glass plate and wet on the top of glass plate. Allow the capillary transfer for a period of 4 to 16 hours. During this period, if filter paper gets wet, change the filter to maintain the capillary action. Remove the paper towel stack. Remove the membrane and rinse with buffer. Check the DNA transfer blotting on the membrane by visualizing the membrane in UV light. Under the UV light, mark the positions of DNA and different lengths. Immobilize the DNA on the membrane with a UV cross-linker with an optimal setting. Transfer of DNA from gel to nitrocellulose membrane. So transfer of DNA from gel to nitrocellulose membrane, what is the principle? So you can do by vacuum transfer methods. Compared to the capillary transfer method, vacuum transfer method is more efficient. Complete transfer of DNA from gel to the nylon membrane takes place in couple of hours compared to 16 to 20 hours in capillary action. Treat the gel as described for capillary method. Cut the nylon membrane and then the silicone mask with opening smaller than the size of the gel. Place the wet Whitman paper on the metal grate. Place the nylon membrane on the Whitman paper and then subsequently put mask. Position the gel on the mask and then place the cover of the vacuum bolt to hold the mask. Apply the vacuum and initiate transfer process. Transfer of DNA from gel to nitrocellulose membrane. Remove the depurination solution and replace it with 20 ml denaturation solution and let the solution permeate through the gel. Replace the denaturation solution with neutralization solution and let the solution permeate through the gel. Again, you have to replace the neutralization solution with buffer and let the solution permeate through the gel. Okay, then you have to remove excess buffer and turn off the vacuum. And now you have to remove gel and check the transfer under UV. Preparation of radio labeled probe. So this is the source DNA. Oligonucleotide primer you have to add for hybridization, and then you have to denature also, then so that those two strands get separated, and then you can do DNA synthesis and clean your fragments and 4 DNTPs you have to add. So this is basically a random primer method. So in this method what happens, a random primer is used to anneal to the template, and then a PCR reaction is performed in the presence of radio labeled nucleotide. After PCR, newly synthesized DNA strand is labeled with radioactive nucleotide. It has following steps. The source double stranded DNA is denatured to generate the single stranded DNA template. A random primer is added and allowed it to anneal to the template strand. It will anneal to the random position throughout the sequence at multiple places. Primer will anneal to the template strand, and now Clino will start the synthesis of two DNA strands. Newly synthesized DNA will give short stretches of multiple level DNA probes. Hybridization and development. So you have to place the membrane in a hybridization tube and pre-hybridize with 10 to 20 ml hybridization solution at 65 degrees Celsius at least for 30 minutes. Preparation of probe for hybridization. Denature the probe by boiling for 10 minutes and quench on ice. Add the probe to the hybridization solution and incubate overnight. Use high temperature for high stringency or moderate temperature for low stringency. Rinse the membrane thoroughly with 10 ml of washing solution. Expose the membrane to an X-ray film with an intensifying strain. This is result after uh, southern blot after hybridization. So the specific sequence band you can see now. If you have any doubt, you can comment down. Hybridization is important because if you want to see your desired band, right, like desired sequence of DNA, so hybridization needed because you are adding some short fragments of like bases which will now go and bind with your specific band, specific sequence of DNA. So that's why hybridization is required. And radio living, if you don't do radio living, how we will then analyze, how we will get to detect the where is the signal? Okay, so this is the importance. Any further question? Harinarayana? Am I audible now? Harinarayana? Okay. So you tell me why you don't need hybridization. What you are doing here? You are trying to detect one specific sequence of DNA or RNA. Okay. So for that you are giving some probes which will go and bind those sequences. Sequences means your desired sequence. So that's why you need to hybridize. Okay. Now to detect the signals whether it is binding or not, you need to have radio labeled DNA probe so that you can do autoradiogram. You can get autoradiogram, right? So this is the need of hybridization and radioactive like probe DNA which will go and bind, like the which will hybridize basically. So this is the need. Okay. Any further doubt, anyone? Harinarayana, hybridization? You don't know what is hybridization, is it? You can speak up if you want to speak up. You don't know what is hybridization? Okay, let me explain. You have one strand of DNA. Okay. You have one strand. So you have a single strand. All bases are here. So you are giving one probe which has complementary bases. So if it is A, T, G, it will be T, A, and C. Now if this probe is going and binding, it will definitely go and bind because these are complementary sequence. So this is called hybridization. Okay. Am I clear? So this is how happening. And now this probe is radio leveled. So that you can detect where your probes are going and binding. Anyone has any doubt? Harinarana, is it clear? Please let me know if you have any doubt. So far we have discussed week 9 tutorial and week 10 tutorial. So we have discussed the tutorial on microscopy, and today we discuss about week 10 content, which is molecular biology part 2, consists of, you know, sequencing and then southern and northern blotting. Next class will be on Tuesday, again 6 to 8 p.m. on the week 11 and week 12 content. So we have two classes left, and we'll be discussing assignments, and if you have any specific doubt, please feel free to discuss. And if you miss any classes, please go through the YouTube videos, and then which the link is there in the NPTEL portal, and follow those tutorial slides, and let me know if you have any specific doubt. So if you don't have any specific questions or doubt, we'll meet on next Tuesday at 6 p.m. to discuss all your doubts, queries related to experimental biotechnology course. Bye bye everyone. Thank you for joining in.